Hello, I'm Dr. Rekebio of the Department of Anatomy and Cell Biology, School of Medical Sciences, University of Cape Coast. I'm pleased to bring to you embryology of the respiratory system. Our objectives are for you to appreciate and understand the normal development of the lungs and its associated structures in the respiratory system. You should also understand why certain congenital anomalies occur and their clinical correlations. As part of the introduction, we are looking at an adult lung, the anterior view of that. So you have this trachea more or less in the midline. That is the right side. That is the left side. So let's start with the right side. We realize that the right side has been divided into lobes of three. So 1, 2, and then 3. With their name as superior lobe, middle lobe, and then inferior lobe. Within the same right lung, you have the main or primary bronchus entering. We will describe it in relation to that of the left. And the primary bronchus will divide into secondary bronchi, which will later also divide into segmental bronchi. Now, when we come to the left side, we realize that the lobes are only two, a very big superior one and then a smaller inferior one. Okay. Now, the division by these fissures have names. So this one in the right lobe is known as the horizontal fissure, and that is the oblique fissure. Then you have the oblique fissure only in the left lung. When we look at the medial view, you are going to have this and that for left and right respectively. So let's take the right hilum. So this region is the hilum where structures enter and leave the lungs. So you have the right bronchi, you have right pulmonary arteries, you have the right superior pulmonary vein, right inferior pulmonary vein. Then you have the mediastinal pleural reflection and then the right pulmonary ligament. Okay, you come to realize that this pleura comes from the visceral layer of the mesoderm. Okay, so when we get there, we look at that. Then you have the left side, you have the left pulmonary artery, you have the left superior pulmonary vein, that's the left main bronchus, left inferior pulmonary vein. Then you have the pleura with its reflection and then its ligaments. Then important to note is the lingula of the left lung, okay, this projection. Then you have a diaphragmatic surface there on both left and right. You also have some cardiac impressions there, and then other impressions of the blood vessels, okay? So you can see that, and then its arching nature. Then you have this and that. You have that as well. So all these will be captured in more details with the gross anatomy, but obviously you know that you have some blood vessels making impressions, okay? Notable ones like the superior and inferior vena cava, the aorta and its arch, and so on and so forth. So how does the lung develop? The first sign of development is the formation of the respiratory diverticulum, which is also known as a laryngotrachea diverticulum. So that is the blue portion you see here. All the yellow portion is endodermal, layers, which will obviously form the GIT. So ventral to the foregut, you have this respiratory diverticulum in blue, also known as the laryngotrachea diverticulum. Initially, the respiratory diverticulum is in open communication with the foregut, but becomes separated by tracheoesophageal folds. All right, so this blue strata is in common communication with the yellow portion, but eventually to be divided as we see. So how is the division going to go about? The tracheoesophageal folds, which are these indentations where the arrow is pointing at, will fuse in the midline here to form the tracheoesophageal septum. So the folds will fuse and form a septum, and that will divide the foregut into the trachea and then esophagus. So here you have the esophagus and have the trachea. All right. So in this very diagram, this will be the anterior portion, and that will be posterior portion because the trachea will come first and then the esophagus is behind. That is why the epiglottis is supposed to cover the trachea to usher food into the esophagus. All right. The opening of the respiratory diverticulum into the foregut becomes the laryngeal inlet. So we use this diagram also to describe almost the same thing we saw in the earlier slide. So yellow GIT, and then that is the respiratory diverticulum. You can see that there is a folding going on there, and then once it bridges, it severs its continuity, and you have a complete esophagus posteriorly, and then a trachea anteriorly, where the lung buds develop. So we call that the bifurcation to form the lung bud because it is dividing into two. The lung bud divides into bronchial bud, which form main or primary lobar, main or primary bronchus, and then you have the secondary or lobar bronchus, and then you have the tertiary or segmental bronchi. Okay. Now we want to see the derivatives of the endoderm, and then also the mesoderm, and then the connective tissue, or if you like, the mesenchyme. The endoderm lining, the laryngotracheal groove, gives rise to the lining epithelium, which makes sense. So the lining epithelium, and that is the tube. So that's the endoderm. Then that is the splanchnic mesoderm. And then you have the glands of the larynx, trachea, and bronchi, also coming from the endoderm. Okay, so you see some glands in the respiratory tract. The connective tissue, cartilage, and smooth muscles are derived from the visceral mesoderm, as well as some contribution of somatomias from pharyngeal arches 4 and 6. So as we go on, know that the area we are looking at is more related to pharyngeal arches 4 and 6, okay, and not pharyngeal arch 1, 2, 3. So this will be a cartilage, that is muscle, smooth muscle, which will become the trachealis muscle that will join the C-shaped complete formed cartilage as the development continues. Then you have a gland there. Obviously, that's a lining epithelium because this is a tube. You have connective tissues as well. Now we want to look at the development of the trachea, bronchi, and then the lung. So the lung or trachea bud will give the trachea. Then you have bronchial bud, which will form main bronchi or the primary bronchi. The clinical significance that when there's an inhaled object, is likely to be found in the right and not the left bronchi. So this is what we are talking about. This is wider, it is shorter, and then it is more inclined vertically compared to the left one, which is longer than that, and then it's also a bit constricted compared to the right one. Then you have the lobar or secondary bronchi. The left and right difference is obviously because these lobar or secondary bronchi enter the lobes. So the name lobar relates to lobes. So that will tell you that if you have two lobes, you are only going to have two lobar bronchi. If you have three lobes, you are going to have three lobar. That is why the left and right differences will be two lobar bronchi for the left and then three for the right respectively. Then you also have segmental bronchi, left and right. The differences you have between 8 to 9 for the left and then 10 for the right. So this is what we are trying to describe. So that is the trachea. You have the two main bronchi or primary bronchi. So this is right main bronchi, that is left main bronchi. Then the green one, which is the left primary bronchus, will now have these generations coming out of it, which will be the yellow ones, okay? And they are the secondary bronchi. So that will be 1, 2, 3, each going into a lobe. So we have the three secondary or lobar bronchi. Then if you compare it to this one, you have only two secondary or lobar bronchi. Then you realize that there's some orange branching also going on. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So those are the segmental bronchi. So 10 in the right, and then you have between 8 and 9 in the left. Usually it is 8. So you have 4 at the upper or superior lobe and 4 in the inferior lobe. There can be some variations though that you end up having about 9 in there. So the bronchopulmonary segment comes about because of these segmental, septations of the bronchus into segmental ones. So we want to compare that. We've already established that you have 10 in the right and then usually 8 in the left. So number 1, 0, 1 is the trachea. Then 0, 2 will be this purple or violet color. That is 0, 2 and then 1, 7. So 0, 2 for the right, which we said is shorter and wider and more inclined vertically. So 0, 2 is main right bronchus, and 17 will be main left bronchus. Then we move to the blue ones. So 0, 4 will be right upper lobar. So we've now got into the secondary branches, okay, or secondary bronchi, or the lobar bronchi. So right, we said there will be 3 in the right one. So we expect to have 0, 4, 0, 5, and then 0, 6. So 0, 4 is right upper lobar, 0, 5 right middle lobar, 0, 6 is right lower lobar bronchus. Then we have only 2 in the left. So that will be 18 and 19. So that is left upper lobar bronchus and the left lower lobar bronchus. Then the yellow ones are the segmental bronchi. So 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16 should give us 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. Then you have 1, 2, 3, 4 in the lower. Then you have 1, 2, 3, 4 in the upper. Basically, that is how you go about it. So for the sequence of lung development, you have by the beginning of the 4th week, in the foregut, you have the laryngotrachea tube, that's the splanchnic mesoderm surrounding it of the lateral plate mesoderm. You have trachea buds or lung buds bifurcating, then it keeps elongating. At this point, the tracheoesophageal septum has already detached the trachea from the esophagus. So you have the bronchial bud by the end of the 4th week. Then trachea bifurcates. You now have several generations ongoing. That is the upper lobe, lower lobe for the left, upper, middle, and lower for right. Some things can go wrong, yes. So during septation by the tracheoesophageal septum, things may not go well. The septation may deviate more anteriorly or posteriorly, or may divert and cause a lot of problems. So here you have the esophagus, which is not continuous with its lower portion. So you have esophageal atresia, that is the trachea. So obviously, if fluid fills up this portion, it's likely to enter the trachea. And you don't want that to happen. In B, you have a continuous esophagus. However, there's a fistula with a trachea. So the trachea joins the esophagus. Then here you have a atresia of the esophagus, yes, but you also have a fistula with the trachea. There's also a fistula here with the trachea and another fistula there from the lower portion of the esophagus. Here is a real photo showing you atresia there, and then you have the trachea there. So that is for the esophagus, esophageal atresia, and that is what the X-ray is also revealing, esophageal atresia. Then here you have the trachea forming a fistula with the esophagus. In this case, the trachea is anterior. For this one, the esophagus was anterior. This is the trachea, which is forming a fistula here with the esophagus. This is synonymous to this one, the B diagram. The larynx is also formed around the region of the formation of the trachea and then the lungs. The lining epithelium of the larynx originates from endoderm, but the cartilages and the muscles are from mesenchyme of the 4th and 6th pharyngeal arches. So remember, I told you that the development of the lungs and its related structures are usually related to the 4th and 6th pharyngeal arches. And these are going to form the thyroid, cricoid, and arytenoid cartilages. The innervation obviously will be by the nerves that are associated with the 4th and 6th pharyngeal arches, which will be the vagus nerve, its superior laryngeal and recurrent laryngeal branches. So we want to follow this development. You have this common tube there. So we are looking at the foregut, primitive foregut there. And then we are looking at the level of the larynx. So these are the lung buds that are forming, as you can see there. Then it bifurcates and continues. But you can see that within this tube are some prominences that are emerging. So those are going to form the epiglottis and then the arytenoid cartilages. So 3 bulges there. And as it continues, you now form this epiglottis, which folds on top of the trachea to direct food into the esophagus, which is posterior. So all things being equal, you are going to have the epiglottis, then the arytenoid swellings. Eventually, everything forms, and then you get this epiglottis, and then you have the arytenoid cartilages. Okay, you also have other cartilages like the corniculate cartilages, and so on. So this is a table that usually gives a very good description of the pharyngeal arches and their derivatives. From the first arch, which is also known as the mandibular, second, which is the hyoid, the third, glossopharyngeal related in terms of nerves, then the fourth and sixth is the vagus and its branches. So our interest today is within this region. So we know that superior laryngeal branch of the vagus and recurrent laryngeal branch of the vagus will do the work for innervation of all the structures related to that region. So for the muscles, you have prepothyroid, you have levator belly palatini, you have constrictors of the pharynx, intrinsic muscles of the larynx, striated muscles of esophagus, then skeletal structures have the thyroid cartilage, cricoid cartilage, arytenoid cartilage, corniculate cartilage, then cuneiform cartilage. The lung develops in stages. So we want to look at the 5 main stages. Sometimes it's shortened to 4 stages when you want to eliminate the first one, which is the embryonic stage. So you have embryonic stage, pseudoglandular stage, canallicular stage, saccular stage, and then the aveolar stage. These are the structures that we expect to see at each stage. Okay, so let's try and correspond them to the structures that are being formed. So for the embryonic stage, you are looking at the human in weeks. This is for the first numbers are for the mice development, that is for human. Okay, and the generations are in terms of the divisions that are going on. So for the human, 3rd to 5th week, so embryonic week, you form bronchi, and you also form the trachea even before the bronchi, but the trachea is not showing. Then the pseudoglandular will form bronchioles up to terminal bronchioles. So you can see that here the pseudoglandular is more or less overlapping some portions of the embryonic stage. But at the end of the day, for the embryonic, you are going to form trachea and bronchi. Pseudoglandular, you form bronchioles to terminal bronchioles. Then canallicular period, you are going to have respiratory bronchioles. And the saccular period, you are going to have the formation of the alveolar ducts and then some circulation going on, or the formation of the primitive alveoli. You have the alveolar stage, which will form the alveolar sacs. Okay, so this will be by the 36th week, okay, of human development. You expect to have everything going on very well. So we can just summarize the stages of development and say that the embryonic period will give rise to trachea, primary bronchi, segmental bronchi. Pseudoglandular will form terminal bronchioles. The canallicular period will form respiratory bronchioles. Saccular period, you have alveoli ducts forming. And then the circulation or formation of primitive alveoli also begin. Then the alveolar period proper, have the alveoli sacs forming. We have to know that all these things are dynamic process and one merges into the other. Okay, so it's for simplicity's sake that we are trying to divide them into these stages. So trachea is there, bronchi, bronchioles, terminal bronchioles. These are telling you of the generation. So here you have 0 generation, here you have 1. So that's where the first bifurcation starts. Then second generation, you are having 2 out of the 1, and it continues all the way. All right. The pleural and pleural cavity, as I mentioned earlier on, that the visceral pleura is coming from the splanchnic or the visceral portion of the lateral mesoderm. So the lateral plate mesoderm divides into 2. You have the visceral splanchnic portion, which goes around the yolk sac, and then you have the somatic portion, which goes towards the body wall. So this is the trachea, which came out of the diverticulum of the foregut, and that is the splanchnic mesoderm around it, because that is closer to the yolk sac and therefore the endoderm. Then that is the somatic mesoderm, which also form a pleura, but then that is going to form the outer pleura, which we call the parietal pleura. So this is what you get. You are going to have the pleural cavity in between the somatic and then the visceral. So here you have again trachea developing within the splanchnic mesoderm, more or less surrounding it, but it's growing into it. You have the somatic mesoderm. So the space between the somatic mesoderm and then the splanchnic mesoderm is the pleural cavity. So that is the visceral pleura. That is the parietal pleura. So if the lung is growing into the coelomic cavity, you have the parietal pleura there, then you have the visceral pleura. So you realize that as this goes on in terms of its growth, it's going to grow into that and going to reduce the space here. All right, so at the end of the day, you see that it grows very big and then fills the whole space and then pushing the visceral pleura towards the parietal pleura. But eventually, you are going to have the visceral pleura investing the lung itself, and then the outer one becomes the parietal pleura. So that is parietal pleura, and then that is the blue one is the visceral pleura. And then you also realize that the space between the visceral and parietal pleura, which is the blue and then the red, is the pleural cavity. Okay, so that is how the formation goes. It can be likened to you pushing your fist into a balloon. So this is an inflated balloon or a rubber. Once you push your fist in, you see that your fist has contact with the surface of the balloon. As you continue to push it inside, you realize that the portion that you first had the touch with your fist continues to move towards the other part of the balloon. So the portion where your fist is in touch with the surface of the balloon, the first portion that's your fist touch, and then as you move towards the other portion of the balloon, you end up having a space which will be like the air within the balloon, separating your fist and the first layer from the outer layer. So the outer layer becomes the parietal, and then where your fist was in touch becomes the visceral pleura. Pneumocyte type 1 and 2 are important layers and cells you find in the lungs. We will talk about that in detail during histology of the respiratory system. But for now, note that you have pneumocyte type 1, which is also the alveolar membrane. Okay, so the alveolus, its membrane is actually the pneumocyte type 1. We will describe the epithelium structure in histology. The pneumocyte type 2 is a specialized cell that produces surfactants. So look out for the importance of surfactant. And when a baby is born prematurely, why surfactant is so important for the survivor. So this is an alveolus, but you can see about 3 sacs, 1, 2, 3. And these are also sacs, 1, 2, 3, 4, 5. So within it, that's what you get. You find the lining of the alveolus itself, which is the pneumocyte type 1, and then you have these specialized cells, which are more of light green. That's the pneumocyte type 2. So the type 1 is the lining of the alveolus. Then you have pneumocyte type 2 interspersed. They produce the surfactant. And that is the epithelium of the duct of the alveolus. I hope this video helps. Thank you very much.